All right. Good morning, good afternoon, and good evening, wherever you are across the globe. Welcome to the November edition of DOHAD Worldwide Webinar Series. I am Dr. Nuruddin Mohammed, Executive Secretary to the International Society for the DOHAD. Today, I have with me Professor Fu Tran, who is the President of the USA DOHAD Society. And along with him, we have today's speaker, Dr. Thea Golden. I would request Professor Fu Tran to introduce our guest speaker, Dr. Thea Golden. And before I do that, the talk is composed of around 30 minutes, and then it will be followed by a Q&A. So if you have any questions to put on, please do so on the chat. So with that, over to you, Fu Tran.
Well, thank you for that, Nuruddin. It is my great pleasure to introduce Dr. Thea Golden as today's speaker. Dr. Golden completed her undergraduate at Stockton University in New Jersey, USA. She completed her doctorate in pharmacology and philosophy at Rutgers University, again in New Jersey. She did her postdoctoral fellowship with Dr. Rebecca Simmons at UPenn. And presently, she is a research assistant professor at the University of Pennsylvania. She has won a number of awards, including the Young Investigator Cassidy Award from the Perinatal Research Society. This is a big neonatal society, and she has been selected as U.S. representative to the recent recent DOHAD Congress. She is a rising star of the U.S. DOHAD Society, as she has served a number of roles in our council. The title of her talk today is Mechanisms Leading to Metabolic Dysfunction in the Offspring Following Gestational Endocrine-Disrupting Chemical Exposure. Please join me to welcome Dr. Golden.
Thank you for that kind introduction. And thanks especially to the organizers of the seminar series for the invitation to present this research project today. I'm really excited because this is a relatively new research project in our laboratory, and it focuses on the gestational exposure to endocrine disrupting chemicals. And I'm grateful we have this period of time at the end of my presentation for discussion, because I'm really interested in hearing what questions you have, what comments you may have about this developing project. So to start, I thought I'd share what the Endocrine Society defines EDCs as: chemicals or mixtures of chemicals that interfere with the way the body's hormones work. And this broad and very inclusive definition, I think, really speaks to just how many chemicals fit this definition and the vast effects that they have. Now, there are several different classes of endocrine disrupting chemicals, some of them probably familiar to you, like phthalates and phenols. And these really are everywhere in our environment, and they're considered ubiquitous. They're found in everyday products like flame retardants, plastics, food, and pesticides in toys. And we are exposed to them through their product manufacturing, the product use, and also the product disposal. And through all these different potential ways of being exposed, we can be exposed through our diet, skin, air, and water. And importantly, NHANES has reported that most adults and children have detectable levels of phenols and phthalates, two of the larger classes of endocrine disrupting chemicals. And EDC toxicity is known and there are suspected mechanisms of toxicity that include hormone-dependent and independent pathways. And the alterations that result from this EDC exposure are some changes to sex organs, such as leading to early puberty and menopause, altered nervous system and neurologic and learning disabilities. Cardiovascular diseases have been linked to EDC toxicity, altered immune function, and a particular area that's near and dear to our hearts are metabolic diseases such as diabetes and obesity. And there is a perinatal window of susceptibility to EDC toxicity in that when there is gestational exposure to these chemicals, we see fetal growth restriction and reduced birth weight, preterm birth, early puberty and alterations to sex organs, and later the development of metabolic, cardiac, and inflammatory diseases. And while metabolic and cardiac diseases have been studied for their developmental origin for some time now, it's really a newer field to consider the developmental origin of immune health and disease. And one of the reasons that this is probably a newer area of research is because until recently, the fetal immune system was considered to be immature. It was thought that the immune system was developing and that upon birth it would actually turn on and be a reactive immune system that we're used to. But really what we've learned over the last few decades is that the immune system is not immature. Instead, it is just functioning differently. It has a function of being a barrier, both the skin and gut, to the in utero environment. And it has a cellular response. It is in fact reduced, but purposely reduced, and not that it is just immature. And some of the ways it's reduced is there's less neutrophil and monocyte chemotaxis, less B cell antibody production, and TLR response. And it achieves this through unique cellular phenotypes. There are some fetal subtypes, such as the B1 B cell and gamma delta T cells or innate lymphoid cells, that are not a prominent population in the adult, but instead are very high in proportion in the fetus. And this helps skew the fetal immune system towards a Th2 or M2 phenotype, which allows for it to be more tolerant of its environment. And the fetal immune system is considered tolerant. And what I mean by that is that when it sees foreign antigen, instead of reacting to that foreign antigen and eliciting an inflammatory response, it actually sees it and it's considered to appreciate that it's foreign but not mount an immune response. And this is true while the fetus is developing, and it's incredibly important because maternal antigens are constantly exposed. The fetus is constantly exposed and recognizing maternal antigens as foreign. And if it were to mount an immune response, this could lead to fetal loss. So over the time of evolution, it's believed that this is something that has been an adaptive evolutionary trait. But upon birth, the immune system shifts towards being responsive to the environment, which is incredibly important because upon birth, the infant is exposed to all sorts of pathogens like viruses and fungi, and if it were to not respond to those antigens as foreign, it could end up having a life-threatening infection. So in order to achieve these divergent phenotypes, the immune system has a layered development of immunity. And there's careful coordination of different immune cell populations, like those I pointed out that are unique to the fetus, like B1 B cells, that come early in fetal development, and then as the fetus develops, the different immune populations develop and take over in dominance as well. And this is achieved through coordinated waves of hematopoiesis. So early in embryologic development, the first immune cells are derived from the yolk sac blood islands. And these are seen in the mouse at gestational age or embryologic day 7, so very early in fetal development. And these actually will go on to seed adult phenotypes such as microglia, which are tissue-resident macrophages of the brain. The second wave of hematopoiesis derived from the hemogenic endothelium of the yolk sac vasculature just a little later, and these immune cells will then go on to derive most of the tissue-resident macrophages. And then finally, there is a third wave of hematopoiesis, which comes from the hemogenic endothelium. This then seeds the fetal liver, which is the main source of immune cells during the fetus's life, and eventually will seed the bone marrow, which is the main source of immune cells in adult life. And these tissue-resident macrophages deriving from these different stages of hematopoiesis during different fetal embryologic days really shows that there's a potential for variability to the permanency of disruption during fetal development. And what I mean by that is that tissue-resident macrophages vary in their lineage. Some organs are considered closed. So the brain, for example, has microglia, the tissue-resident macrophage population, and these, as I pointed out, are derived from that early first wave of hematopoiesis. So in the adult, when there is a need for replenishment of microglia, the microglia actually will self-populate from their in-brain population instead of coming from, say, the blood. There are other organs that are considered open at birth, but these close during the postnatal period. An example of this are macrophages of the lung called alveolar macrophages. And these are considered to be self-populating after that postnatal period from some sort of stem cell within the lung. And finally, there's populations of resident macrophages, such as in the intestine, that are considered open throughout life. So if there is a need for replenishment of intestine resident macrophages, these macrophages will actually come from the bone marrow via the blood. And this is particularly interesting when you consider alterations to fetal development, as the permanency of disruption depends on the tissue population you consider. And finally, I would just like to share that it's become understood that the fetal immune system is, as I said, a tolerant phenotype, but it's also actively participating in organogenesis. And there are some clear pathways that are known in some organs, such as in the lung. We know tissue-resident macrophages participate in alveolarization. If these macrophages do not have their normal phenotype and their normal function, you'll get fewer alveoli. There's also, in the brain, the microglia participate in synapse pruning, and in the kidney, we see branching morphogenesis. And this shows that these immune cells are actively participating in the development of the fetus. So as the immune system shifts from tolerant to reactive in post-birth, this is dependent on the appropriate fetal immune cell development and also dependent on postnatal exposures. And coming from a world where I, in my PhD, studied acute lung injury and really focused on the lung tissue resident macrophages, I was really struck by the lack of understanding and knowledge of pancreatic immune cells when I joined the lab of Dr. Rebecca Simmons. I was really interested in studying the effect of growth restriction on pancreatic immune cells, and in reading the literature, realized that there's very little known about resident immune cells in the pancreas. So we sought out to really characterize the resident pancreatic immune cells in the neonatal rat. So here I'm just showing you a t-SNE plot of single cell RNA-seq data, and this was done on CD45 positive cells isolated from postnatal day 1 rat islets. So CD45 is expressed on most immune cells, so this is isolating all of the immune cells from the islets and then looking at their transcriptome. And what I just would like you to take away is that this is a very dynamic and multi-immune cell population. So there are predominantly macrophages, some T cells and B cells and dendritic cells, and you can see there's some subpopulations to those larger classes of immune cells. And we really wanted to understand the immune populations during the neonatal period. In some organs, it's been shown that the resident immune cells actually are dynamic and are changing during the neonatal period. So we wanted to look across the first 2 weeks of life in both the islets and in the exocrine pancreas. Now, at this really early stage of rat development, there are lymph nodes that are surrounding the pancreas, and those lymph nodes would be captured with our harvest of the pancreas. They're just too small to remove. So when we look at the exocrine immune cells, we're actually looking at both those within the exocrine organ itself and the lymph nodes associated with the pancreas. So first, looking at the populations actually in the islets. So first I have embryologic day 22. This is just before birth, postnatal day 1, 7, and 14. And we really wanted to look at the populations across this 2-week time period. And you'll see in pink, this is the percentage in females, and in black, this is the percentage in males. And what you'll see is that the predominant population in the islets are macrophages. But this decreases with time. As you see, there's an increase in the percentage of T cells. When looking in the exocrine pancreas and the associated lymph nodes, you'll see that again, it is predominantly a macrophage population with significantly more lymphocytes, B cells and T cells in this exocrine pancreas portion. And we were interested in looking at this because there's been a bit of research that has looked at the role of macrophages in islet development. So here I'm just sharing a paper, a body of work that was done using the macrophage deficient mouse. So it's the CSF1 knockout mouse, and this mouse does not have macrophages. And what they did is they histologically assessed the pancreas. And they found that at embryologic day 18.5 in the mouse, this is very late in gestation, just before birth, they found that there is a decrease in the insulin cell mass and the insulin cell size. So these are the beta cells of the pancreas. And that there's also a decrease in insulin proliferation. And when looking at later at postnatal day 21, the non-pregnant adult and in the pregnant adult, they saw that there is a decrease in large islet morphogenesis. So we'll touch on this again later, but there are different sizes to islets. And what they found is that the larger islets were actually reduced in number in the mouse that does not have macrophages. So we began to think about how the pancreatic macrophages may play a role in the development of the pancreas, and really thinking about this neonatal time period where we know that there is remodulation of the pancreas, particularly the islets, that normally happens. And we began to think about the role the macrophages may be playing in that islet remodeling. And in a project that I will very briefly touch on, just to share kind of the framework in which we're thinking about our EDC mixture exposure, we have a established intrauterine growth restriction model. And this is a bilateral uterine artery ligation done on embryologic day 18. So this is actually a surgery model where I go in and tie off the uterine artery, reducing the blood flow to the developing fetuses. And at birth, these pups are born low birth weight. They experience systemic inflammation, which is predominantly M2 and Th2 dominant, and they also show signs of pancreatic inflammation. There's anti-inflammatory macrophages that express high levels of S100A8 and IL-4. And in the adult, we see that they continue to have pancreatic inflammation. There's a decreased islet size, and they have hallmarks of type 2 diabetes, like phenotype. But when we block IL-4, this early X that popped in here, I'm sorry about that. When we did a study where we blocked interleukin-4, what we found was that we actually reduced the pancreatic inflammation, and we reduced the changes to the developing pancreas, that we were able to ameliorate the type 2 diabetic phenotype, such as the increased fasting insulin levels. And this really made us start to think about what is this neonatal period being a real sensitive time point for when there's normal macrophage or normal pancreatic immune cell involvement in the establishment of the pancreas that has lasting effects into adulthood. So in this project, we've asked the question, does gestational exposure to endocrine disrupting chemicals lead to pancreatic inflammation and subsequent type 2 diabetes? And we were very fortunate to be able to collaborate with Dr. Andrea Gore at the University of Texas, who has an established model of gestational exposure to endocrine disrupting chemicals in a mixture. And while it's important and a lot of research has been done on single EDCs, we really wanted to look at an EDC mixture, as this is more representative of the human exposure, and that we are exposed to all sorts of endocrine disrupting chemicals, including the phenols and phthalates and others here that are anti-androgenic. And in this model, she exposed pregnant Sprague-Dawley rats to this EDC mixture daily. It was put onto a little Nilla wafer, which thankfully, like humans, if you put it on a cookie, we will eat it. So the rat ate the cookie that had this mixture on it daily from embryologic day 8 through 18. And then the offspring were assessed. So there was daily weights of the pups for the first up until weaning, and then weekly thereafter. We also were able to get blood from the dams that were exposed. This blood was collected at the time of weaning, so that's 3 weeks after birth. And then the offspring were also assessed at 10 weeks old, and we were fortunate enough to receive the pancreas from these rats so that we could histologically assess their pancreatic phenotype. So first, looking at the birth weights. What we see, there are 4 groups here. So we have females born to dams exposed to the control, and females born to dams exposed to the EDC mixture, and males born to the control and males born to the dams exposed to the mixture. And what you'll see is there's a reduction in the size of the exposed males at postnatal day 1. This persists through postnatal day 7, but by postnatal day 14, it normalizes. And it actually remains normal throughout the 10 weeks. And what this shows is that there is a catch-up growth in these exposed male offspring. And this is really important because we know that catch-up growth is a risk factor even in humans for later metabolic diseases. We also histologically assessed the islets in the pancreas. So here I show you, this is just a pancreas that was stained with insulin by immunohistochemistry. So the brown stain is positive for insulin. And what this shows is the morphology of the islets. And what you'll see is that normally there are varying sizes to the islets. Some are very small, just a few insulin positive cells, and others are very large. And when quantifying the distribution of these islets by their size, what you'll see is that the males that are exposed to the EDC mixture in utero have a loss of these large islets, and they actually are reduced in their overall insulin islet area across the pancreas. I also was interested in looking at the macrophages in the pancreas. If you haven't noticed, that's an interest of mine. And we use CD68 staining in the pancreas by immunohistochemistry and quantified the number of macrophages in the pancreas in several sections across both the section and also throughout the pancreas. And what we found is that there is an increase in the number of macrophages in the males that are exposed to the EDC mixture. And again, I'll remind you that this is offspring at 10 weeks old exposed during their fetal development in utero. And remarkably, they have an increase in their S100A8. This is a particular protein we were interested in looking at because this protein is highly expressed in the macrophages that we see in the pancreas following growth restriction. So we're starting to think about some common pathways that may be resulting from gestational EDC exposure and our surgery bilateral artery ligation model of growth restriction. But really the question that nags us is, how does gestational exposure lead to islet dysfunction and pancreatic inflammation? And some of the things we can assess from the data we have from the collaboration with Dr. Gore are the offspring response. So we see that there is no change in litter size or change in offspring sex. In previous research, we've looked at the effect of BPA directly on fetal islets. So we've exposed fetal islets in culture at varying doses for varying lengths of time, and we don't see the same effect on the islet that we see if it's a gestational exposure. We also were able to assess some of the maternal response. So there was no change in maternal body weight during gestation or through weaning if the dam was exposed to the EDC mixture. And at weaning, 3 weeks post cessation of diet, we were able to measure maternal leptin and insulin. Now this is in the fed state, so in our repeat studies we will be looking at this in a fasting state, but in the fed state we see that there's no change to maternal leptin or insulin. And what this really suggests to us is that there may be an indirect mechanism of toxicity. And an area of research that we are very interested in some of our other pregnancy complications research is the signaling potential of extracellular vesicles. And I'd like to just very briefly introduce extracellular vesicles for anyone who is not familiar with this emerging area of research. And much of what we know about EVs comes from cancer biology research, and that it has been more extensively studied in that field and their role in metastasis and in supporting cancer growth. So generally speaking, a secreting cell packages cargo into an extracellular vesicle. So you have your secreting cell, and via different pathways of biogenesis, the EV is generated, and it may contain everything that's potentially in a cell. So it seems as though if you look for something in an EV that could be in the intracellular environment, it has the potential to be packaged. So this could include nucleic acids like DNA and RNA, proteins, lipids, and even mitochondria, particularly different components of mitochondria and even intact mitochondria in large extracellular vesicles have been demonstrated. And what gets packaged and the size of the EV in that results is really influenced by the pathway of biogenesis and the cell of origin. So once the EVs are released into the extracellular environment, they have the potential to interact with the recipient cell. And these can be taken up by ligand binding or fusion or phagocytosis. And once they're taken up, they have the potential to deliver their cargo, which could have vast effects on the recipient cell. And these different there are different subpopulations of these EVs depending upon what cell type it comes from or what type of biogenesis. And we're particularly interested in looking at these different subpopulations of EVs. The smaller EVs are probably the most well known and understood, and these would be the classic exosomes. Whereas larger EVs, like microvesicles, also are generated primarily through plasma fusion, or plasma membrane fusion. And these vesicles also are lipid membrane bound and are released by the cell, carrying cargo from the cell. So the cargo can vary by subpopulation. We find that there is more mitochondrial DNA and RNA in these larger EVs than in the smaller EV, and that there are different cell types and activities have the type of cell that it comes from, and the activity of that cell will also have a potential to change the type of EVs that is generated and the cargo those EVs carry. And we've developed a mRNA-seq protocol that allows for us to use very low input RNA, because you can imagine there's not much RNA in these EVs. And we've done some research looking at these different subpopulations. So this is just looking at normal, healthy plasma from people. And what we find is that the RNA that we transcribed from these EVs, if you look at this PC plot, those that come from large EVs tend to cluster together, and those from small EVs cluster together. Of course, there's always this one. But what this tells us is that these EVs actually have, depending upon where they come from, what type of biogenesis resulting in either large or small EVs, has an influence on the cargo that they carry. And another major determinant of EVs, of their potential signaling potential, is what cell type they come from. And we are particularly interested in placenta-derived extracellular vesicles because we know in pregnancy that it's been shown that they support a healthy pregnancy. They actually have the potential to signal on the maternal side causing some of the normal alterations we see in cardiovascular system and immune cell changes towards a more maternal tolerant phenotype. And we and others have looked at the role of EVs, circulating EVs, in pregnancy complications such as preterm birth, COVID-19, preeclampsia, and gestational diabetes. And this body of research is really emerging, showing that there is a real signaling potential of these placenta-derived extracellular vesicles, both in normal healthy pregnancy, but also there's a perturbation that exists in pregnancy complications. And we think there's a real utility in studying extracellular vesicles because by looking at the EVs themselves, we're able to begin to understand the pathology. Because the EV secreting cell identifies what cell types are involved in the pathology. The different biogenesis pathways tells us some idea of what the cell is trying to do. And then also by studying the EV cargo, we're getting a glimpse into the cellular microenvironment. And there's the functional potential of EVs that can be studied as well by doing recipient cell functional experiments whereby these EVs are exposed, our cell types of interest are exposed to these EVs in culture, and we can see their functional response. So our platform for studying EVs, we're really interested in characterizing or identifying the secreting cell. We can do this by flow cytometry. Thankfully, flow cytometers have begun to have an increased sensitivity so that we can look at what used to just look like dust in the stream of the flow cytometer. We're now able to actually look at and characterize protein expression on EVs. We're interested in characterizing the biogenesis, both the rate of biogenesis, which we do by looking at the concentration of EVs in circulation, and the pathway of biogenesis, which can be inferred from the size of the EVs. We're interested in the secreting cell activity, and we're using EV cargo to get a glimpse into what that cellular microenvironment is of the secreting cell. As I mentioned, we have RNA-seq protocols we've developed for this, and we're in the process of developing a proteomics protocol that would allow us to use very low input as well. And finally, we're looking at the EV signaling potential via target cell EV exposure experiments. And the reason we're really interested in understanding the EVs in this model is because there's been a considerable amount of research that has shown that extracellular vesicles carry cargo that participate in type 2 diabetes. Now, this is an adult, this is not developmental origin of diabetes necessarily, but what's been found is in a meta-analysis, they found there were more EVs in circulation of diabetic patients. And it's been shown that the circulating EVs actually contain inflammatory and angiogenic proteins that are associated with insulin resistance and beta cell function. And also, I think probably most excitingly, there have been studies where EVs have been taken from obese mice and injected into lean mice into their circulation. And this actually leads to insulin resistance and glucose intolerance in the lean mice that were injected with these EVs, showing that these EVs themselves have a direct effect on the metabolic health of the recipient host. So using the samples that we were given from Dr. Gore, so again, I'll remind you that these are 3 weeks post-exposure at weaning when the blood was taken from the dams. And we isolated EVs from these plasma samples using serial ultracentrifugation so that we were able to capture the larger EVs, the microvesicles, and the smaller EVs by a higher speed spin. And when quantifying the number of EVs in circulation, what we find is that there is an increase in the number of EVs in circulation of the dams that were exposed to the EDC diet. And again, I think this is really remarkable because we are 3 weeks after the cessation of diet has occurred. When looking at the different subpopulations, we see that this is being driven by the small EVs, that there are trending towards more small EVs in the plasma of these EDC exposed dams compared to control, unlike the large EVs, which are more constant in number. And when looking at the size of the EVs, thinking about that pathway of biogenesis, we find that we do not see a change in with EDC exposure in the size of the EVs. We're also very interested in the EV cell origin, and we have developed a flow cytometry protocol so that we could identify different cell populations we think we and others have shown in the literature contribute to the maternal circulating EVs. These include platelets, immune cells, endothelial cells, and trophoblasts from the placenta. And here I'm showing you, we've fully characterized this in some of our human studies. And in the human, the number of, the relative proportion of EVs from these different cell populations are listed here. In this model, the EDC exposure model, I've not fully characterized the EV cell origin yet, but what I have done is looked at the number of EVs that express CD45, and CD45 is expressed on immune cells, and this would suggest that these EVs have come from an immune cell because they themselves express CD45 on their surface. And what you'll see is there's an increase in the number of EVs that are immune cell derived if the dam was exposed to the EDC mixture. So this work is still very much ongoing, and we are continuing to identify the secreting cell populations by flow cytometry, really characterizing the percentage that come from platelets, endothelial cells, and trophoblasts, along with the immune cells that I've shown you today. We're measuring the EV cargo. We are using our RNA-seq protocol, both for polyadenylated RNA and mRNA-seq protocol, and also small RNAs, because small RNAs have been a considerable field of in the literature looking at EVs. We're also very interested in the mitochondrial DNA, and we've done this in some of our human studies and found that the proportion of mitochondrial DNA in the EVs actually is altered in different pregnancy complications as well. And finally, I'm setting up experiments so that we can look at the functional effect of EVs on trophoblasts in culture. And we think that this is very important to study because we know that EDCs and gestational exposure to EDCs has effects on the placenta itself. So when thinking about this from a little bit of a broader perspective, we really think that extracellular vesicles are a window into the health of the pregnancy. And this has potential, it has tremendous potential because maternal circulation is actually an ideal biofluid for biomarker development because it's relatively minimally invasive, and it's something that can be done at many different institutions and hospitals. And what we're really trying to do is use these circulating EVs to potentially identify biomarkers, but really to understand the pathology of what is happening during pregnancy when exposed to something like endocrine disrupting chemicals. And it's our hope that by evaluating the EV signature, we have the potential to discover novel pathways of developmental toxicity. With that, I would just like to take a moment to thank the tremendous group and the support that I have here at the University of Pennsylvania. My postdoc advisor, Dr. Rebecca Simmons, allowed me to spend considerable time on this project. And there have been many in the lab and others at UPenn that have helped with this project. I'd also really like to thank Dr. Andrea Gore at the University of Texas at Austin and her incredible team who had the foresight to collect all of these different organs so that someone like me who's interested in the pancreas was able to really capitalize on a project that she spent a lot of time and money so that we could understand more about how gestational exposure is affecting the entire offspring. And finally, these are the different funding sources that have supported both me, Dr. Simmons, and Dr. Gore. And with that, I will take any questions. Thank you very much.
Thank you, Thea. That was fantastic. So I do have a questions. So what do you think is the driver for sex difference? I mean, why males are affected but not female? And do you think that maybe this involves some sort of sex hormones? And if also the xenobiotic system is affected as well?
So it's a great question. And one of the things that I think is pretty remarkable is that we see a sex effect with our growth restriction model as well as do others, in that males are more susceptible, particularly to metabolic dysfunction. So that's one of the reasons we're thinking that there's a common pathway, is that we're seeing the same sex being altered, whether it be gestational exposure to endocrine disrupting chemicals or a surgery model of growth restriction or several of the other growth restriction models that are used. It's one of the reasons I focus so much on the immune system. We know the immune system between males and females does have some distinct differences, and that could be part of what's driving this. But I think we haven't teased out all of the potential role of direct sex hormone effects as well. So I think it's probably multifactorial, but it is something that we're really considering, especially from an immune lens.
Excellent presentation. Yeah, I have a question for you. Is for future research, would you wish to proceed with checking the levels of EDC in the maternal circulation and try to find out what kind of EDCs were basically there, creating the sort of gestational diabetes kind of a scenario, type 2 diabetes? That's number one. And the second is the intervention or prevention against exposure to the EDCs. And the third question is relationship to neurodevelopment.
Mm-hmm. Okay. So great questions, thank you. So we are actively measuring the levels in both the dam serum and the offspring serum. Now the dams are collected, the blood was only collected at the time of euthanasia of the dams. So it's 3 weeks after the diet was stopped, but they still have, we've measured BPA and PFOS, and both are still elevated in the dams. And the offsprings, the blood was collected at 10 weeks old. So they're very far from their direct exposure. And it is not nearly as high as it is in the dams, but there is an elevation of both BPA and PFOS. These are the only two that we've measured so far, but we are actively doing that now. But more importantly, we're also we're establishing this model here at Penn and collecting these blood samples immediately following birth so that we can see just how high they are. Dr. Gore, when she put this model together, did measure these levels in a subset of animals just to see what the dam levels were. And they were below the no adverse effect levels that are considered safe in the US. So these are levels that should be within a normal human exposure. And not even in the occupational sense, but in the sense of what we see in normal people if you were to just collect blood from you and I. So we are dealing with physiologically relevant doses, but we're actively measuring what they are, with the caveat of them being delayed in how late we're measuring. So there's some clearance, but we know these persist in the body, and we're seeing that in these rodents as well. As for your other question, therapeutic, I would love to be able to start to tease some ideas of therapy. One of the first things that we will do is in our growth restriction model, we've dosed animals with an interleukin-4 neutralizing antibody just in the first week of life, and we're actually able to prevent the development of diabetes. So I plan to inject these rats post-birth for the first week of life and see if we can't reduce that early inflammation that we see in them as well, and then also prevent their changes to their pancreas and probably their beta cell function as well. And then if that's the case, then we're starting to identify a common pathway, something that's probably important to pancreatic development that when perturbed in that neonatal life has a long effect on the offspring's life and presents later in adulthood as diabetes. But there are so many different approaches that have been tried in growth restriction that I think would be of value to try in this model as well. And I'm drawing a blank on your third question. I'm sorry.
And that's the relationship of EDC to neurodevelopment. Does it have any influence?
Yes, so actually that is the area of research that Dr. Gore is really interested in. And she has found that there is some social behavioral changes. Some of the models of, the tests for anxiety and aggression are actually altered in these exposed offspring at 10 weeks old. So that is an area of active research in her research group.
Just to follow up on that lab thoughts, you showed the experiment where they take the exosomes or the EVs from a diabetic rat to the reverse. If you do the reverse, you see if you can rescue some of the pre-diabetic, if you will.
Oh, I see what you mean. So if you were to put EVs isolated from lean animals into an obese to see if, I don't know that that's been done, and that's a really great idea. I guess we'd have to think about how long those EVs would remain in circulation, because you would still have the contribution of the EVs that would be coming from, whether it be the adipocytes in obesity or it be the liver, the hepatocytes, whatever is contributing to those circulating EVs. But if you could outnumber them with the lean EVs and see if they're protective, it's a great idea.
So relate to EV, is there any way to identify the origins of different EVs from different part of the placenta?
So one of the things we're really trying to do, and I'm using flow cytometry, is to identify where the EV came from and using markers like CD45, which is expressed on immune cells. So if I have CD45 on my EV, I know it came from an immune cell. The placenta is really tricky because the only specific cell type to the placenta that doesn't exist on the maternal side is the trophoblast. So I've been using PLAP as a way to identify EVs from trophoblasts, but I won't be able to say with confidence if an EV from an endothelial cell is from the vasculature of the placenta or from the maternal vasculature. So we're starting to use some different fluorescently genetically engineered mice where we're starting to be able to use some GFP labeling of EVs, and we're also really interested in mitochondrial derived EVs. So we have a red mitochondria with a GFP EV. So we're starting to play with that, and I think potentially there's room for us to start labeling. You can imagine having a heterozygous cross so that the fetus is a green GFP EV, and then if mom isn't, then I can start to say that they're coming from the placenta. But that is something that we're working towards. But I think it'll be the only way to really definitively say if these EVs, say from endothelial cells or immune cells or cell types that exist both in the placenta and in the maternal side. Other than the trophoblasts, we're really at a loss to how to tease that apart at this point.
Mm.
And then we're setting up placenta explant as well. And that's something I actually just put into a proposal to the NIH to take placenta from these exposed dams and set up a culture system where I then would be able to isolate the EVs in the cell media. And then I would be able to assess EVs that are derived from the placenta, even if they are from endothelial cells or immune cells. But that's certainly more artificial than being able to do this directly in vivo.
Mm. So one of the things that we found in our research is the sensitive periods if you deal with developmental of different organs. I'm curious if you find the similar results, or if you predict to find similar results if you were to expose these ECDs at different time points. That is like early postnatal period, because that's also another vulnerable period where exposure can change a lot of things.
Absolutely. So my understanding is the original window that was decided, it's embryologic day 8 through 18. She chose to do that so that it didn't affect implantation, so that there wasn't an issue of potential not carrying a litter, and then stopping at day 18 to allow the rats some space to actually go on to deliver their litters. But your point is really well taken, that captures a very large window and certainly doesn't give us any insight into the very end of organogenesis and that late stage or early implantation. So I think it would be wise to tease this apart a bit. The only thing that's so tricky about these chemicals is they stick around so long that if we did something early, it would still likely have an effect through the whole pregnancy. But we certainly could start later and look at the later stages of development. But that early stage, it's going to be pretty complicated in that the chemicals will be still in the system, and we're seeing them 3 weeks after in the dams.
Hey, any questions for Dr. Thea? We haven't seen yet any questions being placed there in the chat box. We're just waiting if anyone has any queries, comments. Questions for you. This is a fantastic presentation and something very innovative we have touched upon. So we just wait till we hear from the audience. So I guess we might tend to finish a bit early, I believe so.
Over
to you, Trent. Yeah.
So while the audience forming the other questions, let me ask you one last question that dear to my heart, which is brain exosomes or EVs. So there's some evidence that shows that they can be identified in circulation, and there's some evidence that suggests that they can also found in maternal blood during pregnancy.
Yes.
Have you find any supporting data for those studies? Or
we haven't looked yet, but you're right. There's a group that I'm drawing a blank on her name. She's down in Texas, and she has been looking at fetal brain-derived exosomes in maternal circulation. And it's incredible research, and it's correlated with brain injury and developmental changes. So it's really, it's remarkable to be able to think that we'll be able to do some kind of, what a biomarker, if you can look at something that's directly from the fetus and is a sign of the pathology. We haven't looked at the fetal-derived EVs that may be in maternal circulation here. However, they may be contributing to the population that we're currently studying. But
Yeah,
I mean, that's a really exciting area.
Yeah, absolutely. I mean, for us, especially organs that are hard to access and, impossible to get to study.
Right. Yes.
Yeah, Laura Gotzo, I
That's
think.
it. Thank you.
At Houston
She presented
Medical.
it at PRS, and it was really remarkable.
Yeah. Wonderful.
Okay, so with that, I believe no more questions for you, Tia. Just a comment that I believe a number of research groups are looking into human maternal circulation, I believe, and also correlating some EDCs with the neurodevelopmental outcome at birth. So probably, you could think of targeting the human population as well, pregnant women, looking into their circulation, the EDC markers, and then correlating with the neurocognitive function at birth.
That would be great.
Yeah, using Pleasanton score or maybe downstream in the early childhood development. Great.
Yes, it's a great idea.
So,
thank you.
So we then close our session for today, and I'm thankful to Dr. Tia for time contribution towards this wonderful talk, and to Trent for being there with us. And for those of you who are not yet the member of the International Society for the DOHAD, please join and take the membership for the International Society. There are great benefits out there. And our last session for this year is going to take place on 7th of December. The presenter is going to be from Doha, Pakistan Society. So you will get the advertisement soon from the platform of the International Society. And we look forward to seeing you again next month. So till then, have a great day ahead. Thank you.
Thank you. Thank you all.
Bye.
Bye-bye.